Say you have a cell. This cell is going to make a protein with the amino acid sequence MET, ARG, THR, ASP, STOP. The protein synthesis is a process by which individual cells build their specific proteins. Within protein synthesis, there are three major steps: transcription, RNA processing, and translation. Before we go any further, let's talk about DNA, also known as deoxyribonucleic acid. DNA takes the shape of a double helix. A double helix is basically just two strands that are combined and create a corkscrew shape, and is basically a ladder. The ladder is made up of nucleotides. A nucleotide has three parts that consist of a sugar, phosphate, and nitrogen base. There are four nitrogen bases: adenine, guanine, thymine, and cytosine. Adenine matches with thymine and guanine matches with cytosine and are held together by a weak hydrogen bond. The sides of a ladder are made up of phosphate molecules and sugar molecules. These are held by a covalent bond and are considered the backbone of DNA. DNA is anti-parallel, which means that they run parallel to each other, but with opposite alignments. This is important when discussing what 5' and 3' are. They are the carbon numbers and DNA ladder sides. The 5' has a phosphate group attached to it, and the 3' ends in a sugar molecule. Enough with DNA. Let's go on to what transcription is. Transcription is the process where RNA molecules are created using the DNA molecules as a template. RNA polymerase is an enzyme that unzips DNA and correctly matches up the appropriate RNA bases with the template. These RNA bases are the same as the nitrogen bases, except uracil replaces thymine. Next is the mRNA processing, the second step. After the RNA polymerase stops after the termination sequence, the coded mRNA is called pre-mRNA. This is because it still contains introns and exons. Introns do not code and exons do. Introns inside the pre-mRNA are cut out or spliced. The product is given a methylated G cap and a poly A tail, and then it is shipped out of the nucleus via nuclear pores and considered mature mRNA. The last step is translation. This process happens in the cytoplasm with the help of the ribosomes. tRNA, also known as transfer RNA, has two main parts: the anticodon and the amino acid. tRNA knows where to start copying a section of DNA when they find the codon AUG. A codon is a three-letter RNA sequence. The tRNA keeps coding until it reaches a stop codon. This process is repeated over and over again, reads in triples, which are known as codons. tRNA matches up with the codon with the anticodon. tRNA finds the appropriate amino acid that corresponds with the codon. After this long process, the amino acids eventually are read by rRNA and creates proteins. A peptide bond is a chemical bond formed between the two molecules when the carboxyl group of one molecule reacts with the amino group of the other molecule, releasing a molecule of water, and they are on amino acids in proteins. There are three sites: the A site, P site, and the E site. The A site is where incoming tRNA with amino acids land. The P site is where amino acids are joined and polypeptides grow. Finally, the E site is where tRNA exits ribosome. The original DNA sequence is TAC, GCA, TGA, CTA, and ATT. The mRNA sequence is AUG, CGU, ACU, GAU, and UAA. Before getting shipped to the cytoplasm and ribosome, they need to get rid of the introns using splicing. Exons stay in the pre-mRNA and gets a cap and tail and is shipped off as mature mRNA. The tRNA finds the AUG and starts coding. The anticodon for the codon sequence is TAC, GCA, TGA, CTA, and ATT. The tRNA keeps going until it reaches UAA, the stop codon. The amino acids MET, ARG, THR, ASP are created to fold a protein with rRNA. Done.